So our next presenter is on her way, and it's Dr. Tania Gendron. And I can see that she's just logging on, so I'm going to introduce her. She is an assistant professor at the Mayo Clinic in Jacksonville, Florida, and has been at the Mayo Clinic there previously as a postdoc and through to assistant professorship, working a lot alongside some prominent ALS research at the area with Dr. Leonard Petruccelli. And she's a Canadian from Ottawa and did her PhD at the National Research Council of Canada prior to that. So she is just about to come on, but you're going to get to hear a lot from Dr. Gendron because she's going to stick with us for our preclinical roundtable panel, which we will do immediately after. And that panel will actually be a prelude to what we're trying to do in terms of podcasts. So since she's still logging on, I'm going to just tell you about what that's going to be. So ultimately what we're going to do is we are going to have a little chat with a couple. Okay, she's ready? Okay, she's ready. All right, so Dr. Gendron, can you hear me?
Yes, I can, and sorry about that. I was in as an attendee.
No problem, no problem at all. We have extra time after, so we can let you do the normal amount of time, no problem. So thank you very much for joining us, and the floor is yours.
Thank you. Thank you so much for giving me the opportunity of sharing my work. I'm not quite sure what the introduction said, but today I'll be talking about biomarkers for a C9 ALS and FTD. And I know that this audience is quite familiar with ALS and the clinical overlap that it does have with frontotemporal dementia and FTD. And in addition to this clinical overlap, we also know that there's neuropathological overlap as well as genetic overlap. Indeed, in 2011, the C9ORF72 repeat expansion was found to be the most common non-genetic cause of both ALS and FTD. And since then, as we've heard, there's been a great deal of effort put towards unraveling how this repeat expansion causes ALS and FTD, and tackling yet another commonality between these two diseases, and that is there really is yet no effective treatment for either. And I do believe that with this discovery, we have uncovered a promising new therapeutic target, at least for this form of ALS. So I know that prior presenters have introduced the C9 mutation, so I'll be very brief about it, but I just wanted to reiterate that the repeat expansion is a hexanucleotide, a G4C2 repeat that's found in a non-coding region of the C9ORF72 gene, or called C9 for short. And because of alternative splicing, it has at least 3 different RNA variants, and 2 of these variants contain the repeat expansion. And then these RNAs go on to encode both a short and a long form of the C9ORF72 protein, which we just heard about. Now, we do know that loss of function of the C9ORF72 protein has been implicated in disease, as we've just heard, and today I'll be talking about the toxicity that might be resulting from the accumulation of repeat-containing RNA transcripts. So again, the repeat expansion can be transcribed both in the sense and antisense direction, and we and others have shown that this leads to the accumulation of RNA into these discrete structures called RNA foci, seen here in these images. And they're seen throughout the neurons and glial cells and other cells throughout the central nervous system of patients. And we've also just heard that these repeat RNAs serve as a template for a very atypical form of translation, and this is called repeat-associated non-ATG translation. It occurs in the absence of a start codon, which is quite unusual, and because of this, it can occur through all potential reading frames. So when you get RAN translation from the sense strands, this produces proteins of repeating dipeptides that encode for PolyGP, PolyGA, and PolyGR proteins. And RAN translation from antisense repeat-containing transcripts give rise to polyPR, polyPA, and again, PolyGP proteins. And we have shown, using antibodies developed for each of these unique dipeptide repeat proteins, that they tend to aggregate in disease, and they form these inclusions throughout the CNS, but found most frequently in the cerebellum, the frontal cortex, and the hippocampus. And among all of these proteins, those that are the most frequently observed are PolyGA and PolyGP. So these key features that I've just described, the RNA foci and the dipeptide repeat proteins, are a result of the accumulation of the repeat RNA, and they're thought to be toxic. So we know that various dipeptide repeat proteins, such as PolyGA, GR, and PR, are toxic in multiple preclinical models. And the repeat RNA itself is thought to be toxic. It can interact with various RNA binding proteins and cause their dysfunction. And as previously mentioned, both the repeat RNA and dipeptide repeat proteins have also been implicated in nucleocytoplasmic transport defects. And so, concentrating on these potential pathomechanisms, the thought is that if you are able to therapeutically neutralize this repeat RNA, either through the use of genetic modifiers, so potentially modifiers that prevent the transcription of the repeat or that prevent its translation, small molecule binders to the RNA that neutralize it, or antisense oligonucleotides that can bind to the repeat RNA and cause its degradation. If you were able to target it in any of those fashions or others, you could essentially prevent downstream events that are initiated by it. And so we think that, as a field, think that this is one of the promising therapeutic avenues that we can approach. C9ASO therapy, I think, is the furthest along in development, with the hope that ASO therapy may go into clinical trial by the end of 2017. So with this in mind, I think it's very important to realize that in the past, many clinical trials for ALS treatments have been hampered by the lack of appropriate biomarkers to help us better design the clinical trial and also to help us better understand the data that we get from it. So for instance, there really is a great need for pharmacodynamic biomarkers to help us confirm target engagement. And one of the reasons this is important is because for those trials that have failed, if you will, we don't know if the therapeutic approach that we were trying was flawed, or whether or not the drug that was being used was just not able to penetrate the brain or find its target. Perhaps we need only to modulate the dose, make a more potent drug, or improve the route of delivery, but the idea in itself is still valid. But without a marker of target engagement, we have no way of discerning this. In addition, we really do need better markers of prognosis and disease course. By and large, ALS is a very rapidly progressing disease, but patients do have quite a heterogeneous population in terms of their survival from disease onset. So if you're trying to detect whether your treatment has an effect on survival, you would need more homogeneous groups to be able to discern this effect without having to include very large numbers of patients, which sometimes is not feasible for ALS. So to investigate this and to bridge the gap in these needs, we first started by asking ourselves if PolyGP proteins, which are easily detected or very abundant in the CNS of C9 expansion carriers, and they're also the product of the repeat RNA that is being therapeutically targeted, could they be used as a marker of target engagement? And we have explored this in numerous preclinical models using small molecules, genetic modifiers, and also antisense oligonucleotides. And for the sake of time, I'll limit it to 2 examples. So here we looked initially in neurons differentiated from iPSCs from patients with a C9 mutation. These were studies that were done in collaboration with the Rothstein group. And we began by determining whether or not we could detect PolyGP and lysates from neurons from patients with a mutation. And to investigate this, we used an immunoassay that I've developed to quantitatively measure PolyGP. And what we found is that we could indeed detect it in cells from patients with a mutation, but patients without the expansion had no PolyGP. We were also equally pleased to see that in the media bathing these cells, we could also detect PolyGP. And we were pleased about this because in this model, we were using media, if you will, as a proxy for cerebrospinal fluid, or CSF. And so we were happy to see that extracellular GPs, like we see in media, correlated well with PolyGP within cells. So we then went on to treat these iPSCs from C9 expansion carriers with an ASO that targets the repeat RNA and causes it to be degraded. And what we found was that upon ASO treatment, we saw a dose-dependent decrease in the levels of repeat-containing RNA. But what's especially good about this is that it had no overall effect on total C9ORF72 mRNA. And in light of the talk, Philip's talk, I think that this is a good finding indeed, that we're not going to be impairing total levels of C9. Consistent with the decrease in repeat RNA, we saw a dose and time-dependent decrease in PolyGP levels detected in media that was collected every 5 days following treatment initiation. And on the 20th day of treatment, cells were collected and lysed, and we measured PolyGP within cells, and again, we saw a dose-dependent decrease in PolyGP within cells. And as we showed above in panel B, we saw a clear, significant association between PolyGP levels in the media and PolyGP levels in the cells. And again, these levels of PolyGP were reflective of how much repeat RNA was present during treatment. So we next investigated this in an in vivo model. We used mice that express an expanded repeat, so a 66 G4C2 repeat. And they're made to express this through the use of adeno-associated viral vectors. And we know that this mouse model develops many of the key features, hallmark features of C9 ALS by 6 months of age. And so we commenced treatment of the C9 ASO or vehicle alone at 4 months and investigated these features 2 months later. So we found was that the C9 ASO therapy decreased the amount of repeat-containing RNA significantly in mice compared to mice receiving only vehicle. And again, the ASO had no effect on endogenous mouse C9ORF72, which is good. And as you'd anticipate with the decrease in repeat RNA, we saw that fewer cells had foci in the ASO treatment group. And this was seen throughout the whole CNS, but we looked at it more quantitatively in the motor cortex, as shown here in panel D. In addition to a decrease in RNA foci, we looked at cells that were immunopositive for dipeptide repeat proteins, so poly-GA, GP, and GR. And as you can see here, there was a clear decrease in cells immunopositive for these proteins by IHC, and we quantified it in panel F. We also looked at poly-GP and poly-GA protein expression by immunoassay using brain homogenates from mice that had been treated or not with the ASO. And again, we see a significant decrease in GA and GP expression. What was perhaps most exciting was that we could detect poly-GP in the CSF of mice, and we also saw a decrease in poly-GP in CSF in mice treated with the C9 ASO. Now, this is probably my favorite slide because what it shows is that CSF poly-GP in these mice correlates with how much poly-GP is in the brain of the mice, how much poly-GA is in the brain of the mice, levels of repeat-containing RNA, and the percentage of cells with RNA foci. So CSF poly-GP seems to be a great predictor of adverse events within the brain that arise as a consequence of the accumulation of repeat RNA. So these findings bode well for the use of poly-GP as a pharmacodynamic biomarker. And so the next step would be to ensure that we can indeed detect GP in CSF from patients. And for the studies I'll be showing from here, I just want to preface it with an extremely collaborative effort to get sufficient amounts of samples to conduct these studies. And we're very happy to say that everyone was really receptive in sharing their samples. This is a collaborative effort using from institutes around the world. And so what we found is that we could indeed detect poly-GP in the CSF from patients with this C9 mutation. And as you'd anticipate, patients without the expansion had no PolyGP. And we were interested to note that both patients with the expansion have the PolyGP, but expansion carriers that are still asymptomatic also seem to have varying degrees of PolyGP. We did detect a trend towards higher levels in patients, but this did not meet significance. In looking at PolyGP levels in samples that were collected longitudinally from the same patients, we noted that PolyGP levels were quite stable over time. And this actually bodes well for its use as a marker of target engagement in a clinical trial, because its stability will allow us better to discern any drug-induced effect on its levels. And we fully anticipate if an ASO is indeed hitting its target and causing the degradation of repeat RNA, we should see a concomitant decrease in PolyGP. However, when we investigated CSF PolyGP levels in our patients and their association with various clinical features, we did not see any significant associations pop out. Certainly, PolyGP levels did not correlate with age at disease onset, nor did it correlate with how long our patients survived following onset. And so based on this, while PolyGP seems to be a promising pharmacodynamic biomarker, it does not provide as much prognostic utility. And so in looking to fill this other gap, we examined whether phosphorylated neurofilament heavy could serve this purpose. So neurofilaments have been investigated as markers of neuronal injury for multiple years now. There is a large body of evidence to support their utility as a biomarker for ALS and other neurodegenerative diseases. For those of you who are less familiar with them, they are intermediate filaments that are in high concentration in axons. They're present in neurons only. And it is thought that as you get axonal injury or neurodegeneration, neurofilaments will be released into the CSF and then into the blood. And the amount of neurofilament released is thought to be a function of how the number of neurons that are affected, the diameter and the length of the axons that are affected, and of course the time course of axonal loss. So in the same population of C9 expansion carriers that we studied for our PolyGP study, we investigated PNFH levels in CSF. And what we found was that patients with ALS, with or without comorbid FTD, had significantly higher PNFH levels than in asymptomatic C9 repeat expansion carriers and also in patients with C9 FTD. And in B, we show that CSF PNFH really has a clear ability to distinguish between patients that are symptomatic and asymptomatic for ALS. But what about PNFH as a prognostic biomarker? We initially looked at its association with a progression score, which was calculated based on the change in their ALS-FRS scores over time. And the ALS-FRS is a functional rating scale, which can be subject to some bias. And what we found was that there was a trend of a correlation between PNFH and our progression score, but it didn't meet our threshold of significance. Nevertheless, what we did observe was a very strong relationship between PNFH levels and survival, and that patients with high levels of PNFH die much earlier than patients with low levels. Now, PNFH is not, as I mentioned, a marker solely for C9 patients. It has been reported to be useful in ALS in general. And when we looked at our non-C9 ALS cases, we found virtually the same data, where there is a clear relationship between PNFH levels and survival. And finally, we looked at PNFH levels in CSF in a longitudinal cohort. And quite like PolyGP, we noted that CSF PNFH levels are also stable across time, especially time periods that would be relevant to clinical trials. And again, we think that this would actually be quite helpful and facilitate the detection of any drug-induced differences in PNFH, for instance, if the drug has a neuroprotective effect. And with that, I would just like to conclude that we do think that PolyGP levels can be used as a pharmacodynamic biomarker, both in preclinical models to screen potential repeat RNA targeting therapies. And we hope that they will also prove useful in clinical trials to confirm target engagement and potentially inform the dose and scheduling of drug treatment. Whereas PNFH levels, which are a strong predictor of survival, we envision that they would facilitate the stratification of patients into more homogeneous groups to better allow us to distinguish treatment effects and the estimation of treatment outcomes. So to finish, I'd like to thank our many collaborators who made these studies possible, and of course my funding sources, and a very big thank you to all the patients and the caregivers who participated in these studies, without whom they would not have been possible. And I'll stop there and take any questions. Thank you.
Thank you very much, Dr. Gendron. Yeah, actually, we're going to go into the panel in a moment, so I'll ask you to stay on the line. But I do have a question relating directly to your talk, and that's great. Actually, I mean, I think it's going to be pretty exciting, regardless of the result for that C9 study, that there's such a strong biomarker presence in that study. You know, not all the ALS studies we've had over the years have had something that works seemingly so well. I have a question in terms of, it was interesting that the pre-symptomatic individuals had sort of varying levels. Some of them had increased levels of PolyGP. And I'm wondering if, let's say the ASO works or some form of C9 therapy works in the future, do you think that maybe PolyGP could be something that could be detected on first symptom? Because I'm assuming that like a lot of the people who would be in the trials, at least the ASOs, will have to have probable or definite ALS. And you know, maybe you'd be able to start treating people very early on if the PolyGP could be a diagnostic biomarker, maybe at first symptom as opposed to. Is there any chance of looking at that, or I don't know?
That's an excellent question. So we do think that the fact you can detect PolyGP in asymptomatic individuals makes that more likely that they could be included in a clinical trial, because it now gives us something to measure. I do not, at this point, believe that it serves as a diagnostic other than confirming whether or not an expansion is, a repeat expansion is present. However, we are conducting multiple studies now where we're collecting blood and CSF from patients from the pre-symptomatic to the symptomatic phases to see at what point we see changes in PolyGP and also in neurofilament. So perhaps once we first start to see a change in neurofilament, that could be indicative of symptom onset, if early symptom onset, and potentially that would then dictate a good time to start treatment, because as you mentioned, the earlier treatment is started, once there are effective treatments available, we believe the better everything will be.
Okay, thank you. A couple questions from listeners really quick. What is the cutoff value for high NFH versus low NFH?
Okay, that's a great question. I'll see if I can't pinpoint it here on the graph. I would have to go back to my notes. I don't want to just randomly shoot it at a number, but it is in our published paper what the great cutoff is for distinguishing between an asymptomatic and a symptomatic patient, or a healthy and an ALS patient. I don't show it here, but another interesting piece of information that came out of our study was, by and large, C9 ALS patients have higher PNFH levels than patients without the mutation.
Okay, and then one last question from the listeners, and then we're going to move to the preclinical panel, is how come C9 does not reduce C9ORF72 total mRNA level, the ASO, sorry. Yeah, sorry.
When our ASOs were being screened, they designed ASOs that target various sequences of the gene. So we have some ASOs that target only the repeat, and some that target around the intronic region. And so there was some careful thought that went into it in finding those ASOs that would have only a negligible, if any, effect on total C9ORF72. And there are 3 variants, and those variants that have the repeat expansion actually are expressed at a lower level than a variant 2. And I think that's also part of the reason why you don't see a big effect on total C9 levels.
Great. Well, thank you very much.